Okay, good afternoon, everyone. We're back in the MARRES Symposium 2026, and we are going to session 2 with Science and Society. This session will be about species and environment relationship. So I would like, as usual, to start thanking your reviewers, so the one who reviewed your papers, including Hubert Bonnefond, who was there just earlier this morning from Darwin Evolution, Florian Bréhin from Créocéan, Cindy Cornet from University of Portsmouth, Emma Alimi from the Met Fund, Stina Kolodzej from Aarhus University, Aziz Moula from Roder Boskovic Institute, and Kayla Plichon from IRCAN Université Côte d'Azur. And today here for your jury, the members of the jury will be donc Steve Como from the Love at Sorbonne Université, Emna Alimi, Aziz Moula, and Kayla Plichon. I will not assess your work, and I will just moderate the questions. So this afternoon we will start with you, Astrid, and Australia. We'll talk about beautiful creatures, apparently. And this log, Raphaël Tremblay will continue also working from the same university, Deakin, in Australia, but more with birds and their environments. And after a break, we will continue with Corinna Lange, who was in Spain, working on some Spanish, also beautiful benthic creatures. And we'll finish with Andrea Vera Espinosa, who was in Portugal, here talking about shark and their environments. Astrid, I'm not asking you how you feel now, it's okay. So if you're ready, not waiting for much more, you can go.
So hello everyone, and thank you for attending my master thesis defense. I'm really strong, so this might be an explanation why there was actually no significant difference. And we did it for every single individual sea slug.
All good. Thank you, Astrid. And there also is saying congratulations. Look at the screen in front of you. And so I would like also take this opportunity to thank your supervisor at Deakin University, Andrew Oxley and Ty Matthews. Yes, thank you. Let's go now to Australia again, Deakin again, but we're going to move to birds and migratory birds. Yes. Am I right? Yes. Migratory birds. Let's have a look to if they migrate.
If they migrate, yeah. Yes.
Good.
C'est les flèches, hein, c'est ça?
Les flèches. Yeah, you want to try?
Yes, perfect. Okay, hello everyone. Thank you for being here. I'll be presenting my master thesis where I looked at the interannual variations in short-tailed shearwaters during their non-breeding period. So first of all, seabirds are unfortunately one of the most threatened birds globally. So seabirds are birds that share their time from breeding on land and feeding in the ocean. And their global abundance actually decreased 25% between 1950 and 2010. And as we know, the ocean is changing, it's warming, the climate is changing. So this decrease in abundance probably is even higher now since 2010. So these birds are especially vulnerable to fisheries bycatch, but also to environmental drivers such as their habitat shifting because of climate or their prey redistributing. And these threats are especially true for transhemispheric migrants. So these are seabirds that migrate across hemispheres. So these are seabirds that will go from one pole to the next pole to follow resources for feeding and further survival. And this brings me to my species of focus, the short-tailed shearwaters. And they are transhemispheric migrant seabirds. They're one of the most abundant seabirds in Australia. So they represent a really huge biomass. And when they migrate and they feed in the ocean, they take a lot of the resources from that ocean so they can act as ecosystem engineers in that environment. However, their population is declining for similar reasons as I mentioned just before. So these are birds that are really dependent on the environment and environmental variables for their survival. This is why it makes studying how they respond and adapt to this change in environment interesting. So these birds carry one of the longest. Okay, yes. They carry one of the longest migrations in seabirds. They will spend their breeding period in the southeast of Australia over here towards Victoria, where they'll have their chicks. And they will start the non-breeding period, leaving the colony and spending a couple days in the Southern Ocean here, feeding and building up reserve for this really lengthy migration, where they'll then make it to the Northern Hemisphere, where they will spend their overwintering phase during the boreal summer. They'll spend a couple months there feeding and foraging. And then they will migrate back down to the breeding colony for the next breeding season. So these phases are interconnected, and what happens in one phase in the non-breeding period might impact what happens in a following phase of that period, and eventually impact the following breeding season. So it's important, it can have possible carryover effects. So it's important to look at all these phases together to see where there's room for flexibility, or maybe room for, or where there may be less room for this flexibility. We know that these seabirds migrate. We know their overall tracks and timing of migration, but there isn't a lot about how this migration and the timing of this migration is maybe changing as the climate and the ocean is also changing. So this brings me to my main two aims. For the purpose of this presentation, I'll only be focusing on the first aim. However, my second one is explored in my written thesis, and I'll be more than happy to answer any questions about it afterwards. So the first aim was to investigate interannual trends in the timing, the spatial extent, and the duration of the phases of the non-breeding period, and also looking at any potential carryover effects connecting the phases of the non-breeding period. So to do this, we use data from GLS devices, which are really tiny and very light devices that we put on the tarsus of the bird. And these devices measure light. So if you know the timing of sunrise and of sunset, you can know where you are in the world. So these devices provided us with the geolocation data, which allowed us to map out the birds and ultimately have an idea of the timing of where they are and when. And so we collected data from, for the purpose of this study, we only used data from a single colony in Gable Island in the southeast of Australia. There's data from 59 birds over a period of 8 years. I do want to preface that so each bird was only sampled over a single season, so only over 1 year of data per bird. That not all the birds, because of failure with GLS data processing, we don't have the data for the whole non-breeding period for every bird. So maybe for some birds, we'll only have the beginning, the northern migration. We won't have the return back. So this is important to consider when considering the significance of the statistics, because there's a lot of variation in sample sizes between phases and between years. So going into the results, starting with the first phase of their non-breeding period, called the pre-migration phase. So this is when they've left the colony for the last time. They're done raising the chick, and they'll spend a couple days in here feeding and building this reserve for the lengthy migration. And I looked at the duration in days over the study period of this phase, and I noticed that across the study period, this phase actually progressively decreased. And then I looked at the date at which they left the colony, so the start date of this phase, and I saw that the birds were progressively leaving the colony progressively later. So this might mean that they're taking actually longer to raise their chicks because they're spending more time in the breeding period, which might imply that there's a decrease in food availability during this period, because they'll go and they'll feed south of Australia and closer to Antarctica. And there might be a progressive shift in the productivity, meaning they might have to travel more, or it might be harder for them to find suitable prey during this breeding period. And this causes a decrease in this pre-migration period, which is an important period for them to build reserve and have the energy required for this migration. So this might mean that they have less time to regain condition before starting this migration, which brings me to the migration, the second phase of this non-breeding period, which is the northern migration. This is a very short phase that happens over 2 weeks, where the birds will cross the southern hemisphere to the northern hemisphere, where they reach their feeding grounds. And this phase, so I looked at the duration of the phase, as well as any interannual trends in the start and end date of the phase, and noticed no trends. So this would mean that this is a very fixed and very stable phase, which could imply that this phase implies photoperiod control, meaning that the cue for the start and end of the phase is driven by light availability, and is something that's more hardwired inside the evolution of the bird. And so the birds are leaving when to match a seasonal productivity window in the northern Pacific, because the birds will actually start their northern migration when the productivity in the northern Pacific is greater than the one in the southern Pacific. And still in this phase, I looked at at which latitude they finished the migration. So over here you can see they're progressively ending the migration further north, which would be consistent with the fact that the pole world redistribution of the prey, as the sea ice is warming, the sea ice is receding, the prey is following the edge of the sea ice, which would mean that maybe they're ending their migration further north to end it where the prey is more available. And now looking at the overwintering phase. So this is a really a longer phase where the birds will spend time traveling and feeding, following prey for a couple months in the northern hemisphere here. And looking at the duration of this phase, there is a slight but still significant increase in the length of this phase, which would mean that birds are spending more time in the northern hemisphere during the boreal summer, meaning that it might be because, progressively during this phase, birds are actually traveling further north to follow the movement of the prey, which would maybe indicate that they're having to travel more. And this increase in duration of the phase might also reflect the decrease in body condition, because if they're traveling more, they're spending more energy. Maybe it's harder to find prey, meaning that they might not have an optimal body condition for their southern migration, return to colony. Which brings me to the last phase of the non-breeding period, which is the southern migration. So this is when birds leave the feeding grounds to return back to the breeding colony for the breeding season. And similarly to the northern migration, this is a very stable and very fixed phase. So I saw no interannual trends in phase duration or timing of the phase, which would indicate that it implies photoperiod control as well. But as we saw, there is an increase in the phase of the overwintering phase. So this increase in overwintering phase that may lead to a decrease in body condition, and the fact that the southern migration is very stable and fixed, puts a lot of pressure on the bird to return back to the colony by a certain date because of photoperiod cues, meaning that maybe some birds might actually not make it back to the breeding colony and might actually die before the start of this southern return. I do want to make a note that this is a biased sample, because the birds that we're studying and we're looking at in this study are birds that we know survived and made it back to the colony, because we were able to retrieve this GLS tag from them to be able to process the data. So we don't know the movement or the behavior of birds that may have died or may have not made it back to the colony for breeding season. And I also do want to mention that we saw, as I said, an increase in this overwintering phase, but both migration phases are very stable and very fixed. So how can we explain that? Would be probably because of varying sample sizes between these phases, as I had mentioned at the start. An overwintering phase had very low sample sizes, especially for some years. There were only 2 birds, for example. And it could also be explained by the fact that this increase in duration is slight, though it is significant. And it might just be that it's taking a bit from the northern migration, a bit from the southern migration, but not enough to then see any visible, statistically significant trends in both of the migration phases. And then looking more on a carryover effect, so connecting these different phases on an individual level, I noticed that birds that left the colony later actually started their southern migration later and ultimately returned to the colony later. So this might have potential fitness consequences on the bird, because birds that return to the colony later might have less of an easy access to mates, less access to burrows where they can make their nests, and might miss the overall breeding synchrony window. However, this cannot be confirmed at all because we didn't monitor the breeding success of the birds, but this would be a really interesting future study to connect the non-breeding period with the following breeding season. So in conclusion, we saw that both of these migration phases are very fixed phases, very stable phases that both imply photoperiod control. And this, coupled with a changing ocean and changing environment, might mean that birds might have to undergo longer migrations to reach more optimal foraging areas during the overwintering season, for example, and which might mean that then they need to travel more, or which might lead to lower body condition and ultimately leading to less birds returning back for the following breeding season. Future directions that would be really interesting would be to connect these phases a bit more directly with environmental conditions. So connecting more, looking at every year how the environmental conditions change and connecting it with the trends that we see in a more explicit manner. Next, as I just mentioned, monitoring the breeding success, seeing if there's any connections, any potential carryover effects here. And lastly, to add any other colonies. As I mentioned, these are really abundant birds in southeastern Australia. And so there are many other colonies, and it'd be really interesting to look at data and sample other colonies to see if these trends are also seen in other colonies, and if there's any possible colony effect here. And I want to end by saying that the more I research this, the more I realize how complex and how many factors actually really go into this cycle of the bird's life, and how this study only provides a very small and narrow window into explaining what drives these different phases and what drives this overall pattern. So thank you to my supervisor, and thank you to all for listening.
Thank you, Raphael. So we're going for the question on the other way around, starting with Kayla and Aziz.
Thank you for your presentation. It's a lot of work, and congrats on that. Also, you made it very clear how you put everything into place, so congrats for the presentation. If I understood correctly, you said that the 59 birds were only tracked once.
Yes.
So you only follow, so you have the data for the bird only for 1 year.
For 1 year, yes.
Okay. In that case, can you separate the real temporal effect from just the random sampling that you happen to have?
So you're asking, like, if I differentiate the fact that it's different birds, different individuals every year, or
how
yes?
can you make differences? How can you separate the potential confounding effect that you have individual variants and temporal variants?
Yeah, so I did a lot of different modeling throughout it, and I did look at individual as a factor, and I didn't see any significance in differences in individuals between the years when analyzing my models. Yeah.
And do you have an idea of a protocol you can follow to actually track and make the difference between the two of them?
Well, it would be very interesting to track the same individual for over multiple years. But I think considering that it's hard because we need to retrieve the GLS device every year, and if we stay in the same scope of keeping different individuals per year, I would think having a more complex model that considers all these different factors together to consider for the possible individual effect would be a good step to do in the future.
And do you know if there's other ways of monitoring the same birth over the years?
Yeah, so I mean, it would just require more human effort to find the same birds because they don't have always the same nest. But otherwise, maybe using another device that's not a GLS device that could be monitored over a longer period of time. That maybe you don't have to retrieve the device to be able to process the data. So yeah, I'd probably just say using a different tracking device.
Thank you. I think, yeah, and my last little question is more an open question. How would you limit, because you said, and then you have a survival bias, because you only follow, but it's kind of the same history. How can you limit this bias?
Yeah, kind of going back to my previous answer would be to use a different device that wouldn't require you to retrieve the tag to be able to retrieve the data. I'm thinking of maybe some kind of GPS device that might send the data to the cloud. So maybe you'll be able to get data from a bird that might not make it back to the colony.
Okay, thank you.
Michaela, so we're going to Aziz and then Emna.
Good talk. Just to Kayla's point, like, are there any other ways to observe this? I mean, I don't know about birds, but like we know the tracking.
Yeah.
So just look up, like, and is there no way to calculate the abundance from flight rather than? Because I mean, 59 individuals is not enough, and for this you wouldn't need individuals.
Yeah.
Because there's a population movement.
Yeah. So yeah, but they're all from the same population in this case. But there are previous studies that looked at that they're using data from fishermen reports and from visual sightings, not just fishermen, but just overall visual sightings. Yeah, that does provide a lot of interesting data. But obviously, when you're tracking such a big movement, it's hard to only rely on visual data. But for sure, tracking their behavior or overall movement during the overwintering period in the northern Pacific, it would be interesting, to use observer kind of.
And then in your introduction, you said the most abundant, but the population's declining.
Yeah.
What have we got to detect this?
Well, it's monitoring populations through the different colonies that are in the southeastern Australia. So these are populations that's been studied for a lot of years. As we can see, we have 8 years of data. So they go there every year to sample and tag, not just in this colony, but also other colonies. So it's a part of an ongoing long-term monitoring.
Okay. So we have 8 years of data, but actually each year is just one data set.
Yeah.
So obviously it's not enough to assess the impacts of climate change. But do you think, can you just go back to your GLM?
Yes. Wait, which one?
Yeah, this one. Did you try any other models on this apart from a linear one?
I did, but was struggling quite a bit with it. Because,
Maybe it's oscillating.
Yeah, and I mean, there is also a lot of variation in the sample sizes, and these are intraannual trends and not necessarily intraannual variations, because that would.
Are the others the same? Yeah.
Oops. Yeah.
Yeah, maybe you need like a non-linear regression or a GAM, maybe. Yeah. Get R out. Okay, and final question. So the movement is caused by prey, by where they go to, right? So, is there historical data on this? Like, so you said in the northern hemisphere that we can see like some outliers and stuff like, and maybe they're moving closer to the pole, correct? Yeah, but would we expect this in the south as well?
Yeah, so in the south as well, obviously they're spending less time in the south, and the tracks that you see here, it's only from the non-breeding period, but they go here and forage here all during the breeding period as well. And yeah, like I didn't look at the distance of the trips during the breeding period, but as you can see, it is very close to the ice edge as well. Is that the productivity really closer to the coast near Bass Strait isn't as high as it is more south.
So this is a new kind of recent trend that they are going.
Yeah, yeah, it is. Yeah.
Okay, thank you.
So him now. It's fine.
Thank you. Thank you for your presentation. Some of the questions of Aziz answered already. You were answering already, so I won't tell them again. Maybe my first question, you mentioned that birds can be ecosystem engineers in your introduction. Can you develop that a little bit more?
Yeah.
How?
Yeah, so when they migrate, they'll all follow and leave it around the same couple of days. So this represents a very big biomass. If you see birds as a population, not as one individual, it's it could be considered as a megafauna species because they'll all go and feed at the same time. So take resources from the ocean at once, which can act as ecosystem engineering because they have an impact because they arrive in such large numbers and abundant numbers.
Okay. And you spoke also about feeding.
Yeah.
Do we have an idea of which preys they take? Whether I'm wondering, like it's a large area, are they the same more or less?
So it varies. It varies with the season. So they feed from everything between krill to fish to squid. And there is literature that reports a shift in their prey and their diet as the season progresses. So they are adapting the type of fish they prey on or the type of food they'll prey on depending on the productivity and the availability.
Okay. And a methodology question, were the tagged individuals all adults or there were sub-adults or?
So all the birds that were tagged were birds that had successfully breeded the season before. Okay. So they were birds that had chicks when tagged,
Thank you.
Thank you. So that leaves the floor for you.
No, I remember. Yeah. No, it was very. I think like Aziz mentioned, like maybe there would be some oscillation or something like that.
Yeah.
Do you have any idea what could explain that?
So, quick answer would be very varying sample sizes, because. So, for example, some years I only had 2 birds, so it's kind of hard to get a general idea of what the trend would actually look like. If not, like obviously 8 years is not so much to see a very visible trend. So certain years, for example, over here, we don't have 2019 because there was a mass ocean warming event. So a lot of birds died and we weren't able to retrieve the GLS data. So there is variation from year to year that we can't necessarily see on over 8 years. So I would maybe explain it just by some years having slightly different environmental conditions than others.
Okay, so you didn't try to see if like any new year, for instance, would have.
So yeah, so that's a future direction that I wanted to go into, but in this case I did not know.
Okay, yeah, and because I think you should also not, the variability could also be by itself something interesting to be like from one year to another. Some years all the birds are living together, other years there's a big range you can see on your data, and I think that's something that maybe should be investigated.
Yeah, I agree. Yeah.
And I was also wondering, is there data on those birds if they also rely a lot on fishing activities, like human fishing activities? Because we know that populations of birds finally are artificially high because they're relying on humans almost to feed.
Yeah.
Also the case for this species, or not?
I'm actually not entirely sure. I would, just from how it is the case for many other seabirds, I would guess yes. However, seeing how actually, I would say yes, because a lot of birds are following the prey that's moving further north. But as part of my thesis that I didn't mention here, I looked at the different clusters of where they spend their overwintering period, and some will stay still, like they wouldn't enter the Arctic Circle, for example, and they'll stay closer to Japan, and I could hypothesize that they're following more fishing vessels to get prey. So yeah.
A last question. You say at some point that maybe if they stay longer in the north, they would not have energy and they would die up there. Is there any observation of that, or it's just a pure hypothesis?
Currently, no, it's just a pure hypothesis, because it's all trends that are kind of somewhat new, because this shift further north and all that is more of a newer observation. But yeah, and it's a pure hypothesis. Yeah.
And because you start with the hypothesis that they are going to suffer from climate change a lot.
Yeah.
But since they feed in the pole, and we know that the Arctic Ocean and the Southern Ocean are becoming actually more productive.
Yeah.
So maybe they could benefit.
Yeah, they could, but the only thing that I could think of is that it would just require more energy for them to have to travel more, and therefore build more reserve before doing this migration. So they'd have to adapt that way. Shift, but that's what's happening to a lot of seabirds. There's a paper that came out like 2 months ago, I think, that talks about the effect of climate change on all these seabirds, and they're seeing that they're just redistributing, because these are birds that physiologically are already pushed to very extremes. So the way they're adapting is by changing their distribution and extending it. Yeah.
Are we talking about birds?
Seabirds.
Yes, seabirds. I mean, but individuals or the populations, like natural selections, or
the what? Are
we talking about individuals relocating themselves, or populations?
Populations.
So by natural selection.
Yeah.
For generations. Yeah, sometimes I'm curious too.
Just one more question about the carryover effect.
Yeah.
Where did this data come from?
From this data set.
The 59 birds.
The 59 birds.
So what proportion of birds went late and came back late?
Yeah. Is that your question? Or no?
What proportion?
The numbers of it. So this is not comparing the years. It's comparing. I didn't put it here. It's written in my thesis.
You're not gonna read that.
Yeah, no, it's okay. No, it's it was. I did a linear model looking at so there was a greater proportion of birds returning. So the later they left the colony, the later they returned to the colony. I don't have the number exactly in my head. Sorry.
It must have been very low.
What do you mean? As in, because it was a small number of birds. Yeah.
Okay. I think I'm not hurting the feelings of anyone. If I give back the floor to you.
Yes.
How does the tagging work? Because I know some years we have less than others, but if because you all started, did every year start with the same number of tags and they just didn't come back? Or did it depend on how many birds you were?
So it's a combination of factors. So obviously, it depends. Like we try to tag the same number every year, having them tagging the most possible. However, it depends on if maybe the bird might come back later, the bird might die, the bird might lose the tag. So there's a lot of tags that are lost. So that's why the data isn't always super high. And some individuals, there's 2 individuals. Some years there's 2 individuals, others there's 14 individuals, right? So it's a hard thing when you rely on retrieving a device like that.
So every year you just at the beginning, right?
Yeah.
You just tag as many as you could.
Yeah. So it varied. I think the number. I believe it was around like 20. I know when I went, we tagged around 20 individuals. It takes time because you need to find individuals that are breeding and have chicks, and yeah. Karina.
It seems like you could learn a lot from the environmental conditions. That
Yeah.
is one of your conclusions.
Yeah.
And what do you think how this could be done? Because I mean, now I guess the only thing that you have is literature, but how, can you include like the seas, the temperature,
So I did actually extract a lot of environmental variables through just remote sensing data that's available online. So if you want more of a small scale and precise scale environmental variables, that would be a way to connect it with environmental conditions. It is something that was kind of the goal of my thesis also, was to connect it with all these variables that I extracted, but because of time constraints, I chose not to focus exactly on that right away. But yeah, it's doable. Yes, absolutely, yes. Jakob?
When you retrieved the device, did you give the bird a new device, or was it all new birds?
It's new birds, because the devices are retrieved in around like our fall here, if you want, like when they come back, so September, October. But the birds are tagged later, so they're tagged in February. So it's not the same. You're not tagging at the same time that you're retrieving the device, because you have to let them breed. Because you retrieve the tag before right when they get back to the colony, and then breeding season happens, and then you'll tag them. Well, it's because we want to tag birds that have successfully breeded. So we want to know that they have a chick by the time we tag them. Sophia?
I was wondering, because like the migration is so depending on their diet, is there any study done on the feces of the bird?
To analyze what they've eaten and everything?
Yeah, in like both locations.
Yeah, not that I know of, but it's a very interesting question, actually. I'm curious about that. Yeah. But there is, I think, on actually, I'll come back to that after. Yes.
I just have like a quick question. How big are these
birds?
Yeah, no, the birds, like the group of birds. How many of them?
Oh, the colony.
They move like they're a huge mass.
Yeah, they're a lot. The colony that I was in Gable Island, I don't even know how many birds there were, but I know. But when we were on the field, it's like you could hear them more than you can see them, because we work at nighttime. And when I think of, I think that there are a couple millions actually in the southeast of Australia. So not just the colony that I worked with, but yeah, a lot.
What about one bird then?
One bird, how many? They're like this. They can like, they're not, but their wings, they're not huge birds.
Yeah, we have time for more questions.
Ocean,
sorry, it's a quick question, but do the different colonies also move around at the same time? Do you know?
Yes. Yeah.
I had a question about slide 24. Can you just kind of re-explain, because that was pretty fast and it was interesting.
Yeah, so they're ending the migration further north, meaning that when they stop flying and they're like, okay, we made it to the overwintering grounds, let's start feeding. It's ending progressively north, which I thought that was consistent with the fact that the prey is also moving further north.
What do they feed on?
So as I mentioned, it was krill, fish, squid, just a bunch of different things.
And just on that, do you quantify kind of kilometers per year or something like that?
Of how long they travel?
Yeah, more.
Oh, how further north?
Yeah.
For this? No, for this I did not do, though I have the data for it and I could do it. But I did not do it. But yeah, no, yeah. But you can see like the difference in the latitude, the degrees of the latitude.
That's actually the slope of your slope. Yeah.
Yeah.
Two more questions.
Yeah.
So why not just put a few GPS tags that would solve many of the mysteries?
Yeah. So, what do you mean? As in, like, to the mysteries of the ones that don't get it back and stuff? Yeah. So we do put GPS tags on a lot of them. However, the one that we put, I believe the life of the GPS is a bit shorter. There's a lot of devices out there, a lot of different technologies with different lifespan. But yeah, it's a different kind of data that you collect from it. Yeah. I could have. It would have been like a whole other ballpark, but I could have, yeah.
And the last question.
Yeah, I was just wondering what the conservation implications of your result for me, because I don't really know a lot about how could you protect seabirds, and also knowing that migratory species are into.
That was also a question online.
That's a great question. I mean, obviously, like a lot of the impact that we're seeing on birds is on a very large scale. So the ocean warming and all that, and as an individual, how do you stop the ocean from warming? But obviously, I mean, we've all seen these pictures of birds with like plastic and all that. So I do think in terms of managing trash and all that, you could have an impact on seabirds and keeping the ocean clean. But if not, I do think, like for example, this colony it's an island that's owned by the state of Victoria, so it's an island that no one can really go on unless they have like a pass to go on everything. So this does help them because it's where they breed, and it's where so protecting also the breeding grounds, I think, does have a big impact on their survival and their conservation. Yeah.
Thank you. Congratulations. You have a few congratulations if you want to check also.
Thank you.
One of you.
Thank you.
And the question. So congratulations. We are now moving for a break, and we are coming back here at 3:45. The next presentation will start at 3:50 in the room at 3:45. 30 minutes. See you in 30 minutes, Kayla.
Gudas didn't start the timer yet.
Yeah. Okay. So I think we're good. Just to check, Kayla. Kayla, can you hear me correctly? Seems good. Okay. I think we can go. So we are back for the second part of the session with species and their environment. We'll have Corina and Andrea. Corina, I think you can start your 15 minutes timer. It's all good.
Great. Welcome back, everyone. I hope you're excited to learn about this beautiful coral and, as Christoph said yesterday, about boring algae. I wrote my thesis.
I'm really sorry, Karina. Your joke was very fun. I have to stop you at that because we have another problem. Just that I see. Yeah, I know. It's not about the joke, sorry.
Sure. You're cutting it, aren't you?
I don't know what it's. Where? Okay, very good.
Great. So I wrote my thesis at the Spanish Institute of Oceanography in Santander on spatial risk assessment and conservation strategies for benthic habitats along the Spanish north coast. The study area is, no, this is not. Yeah, the study area is the Spanish North Atlantic Marine Demarcation, in short NOR, that reaches from the French to the Portuguese border. It has a narrow shelf of 30 to 50 kilometers wide and a pronounced shelf break at around 170 meters depth. The coast is also known for its high wave exposure, mesotidal conditions, and seasonal upwellings. The result is a highly productive and diverse marine environment. For example, the rocky coast, the rocky shelf provides suitable habitats for coral gardens formed by Dendrophilia conifera, and the sheltered rias in Galicia host the only male beds in the NOR. These two habitats were selected within the larger BioDiff project for their high ecological value and vulnerability, with the goal to increase knowledge and provide conservation recommendations to the Spanish government. Dendrophilia conifera is a slow-growing cold-water coral distributed along the whole coast. It creates complex reef habitat and provides biodiversity, mainly on rocks, but sometimes the smallest hard piece within otherwise soft sediments can be enough for individuals to settle. Male beds, not so boring after all, are biogenic habitat formed by free-living coralline red algae. They consist of millions of calcified small nodules that accumulate on the seafloor and thereby also create a structurally complex habitat, supporting high biodiversity and providing nursery areas for many species. Both habitats are recognized under several European and regional conservation frameworks, including the EU Habitats Directive, the Marine Strategy Framework Directive, the Nature Restoration Regulation, and OSPAR. However, these frameworks are often broad and habitat independent. At the same time, managers face limited habitat information, multiple overlapping human pressures, and uncertainty about how specific pressures affect specific habitats. And this creates a need for more spatially and habitat-specific assessments. Therefore, I wanted to know how and where these habitats overlap with anthropogenic pressures, how this translates into risk, and what it means in terms of conservation. For Dendrophilia conifera, the main pressure is bottom contact fishing. Therefore, I focused on three gear types, which are bottom trawling, bottom set gill nets, and bottom long lines. As you probably know, bottom trawling causes the highest physical disturbance because the net is continuously dragged across the seafloor. To not break the nets, they only operate on soft sediments. It was therefore included in my analysis as precautionary approach. Static gears are less impactful, but because they operate on rocky bottom, they can still significantly damage corals by entangling or even breaking them, especially the gill nets during their retrieval. Important to note is also that more than 90% of the vessels in the NOR are part of the small-scale fleet that is not represented in VMS data that I used. because gillnets and longlines are very common in this fleet, static gear effort is underrepresented in my data, whereas bottom trawling is largely captured. Mailbeds, on the other hand, face several overlapping, several coastal pressures because the Galician rias have always supported high-intensive human use. The main pressure are mussel farms, locally called bateas, because there are literally thousands of them, of those platforms that cause sedimentation and organic enrichment. In my analysis, I included these other pressures that I found relevant, but in this presentation, I'm focusing only on the bateas and on the cumulative pressure analysis of all these pressures. If you are interested, I can show the other result maps later. Next, I overlapped the habitat layers with all of these pressure layers. And because the habitat, the pressures, and the available data differed, my analytical approaches also slightly differed. For Dendrophilia conifera, we had a species distribution model available that allowed for a more detailed risk assessment, where for male beds, I followed this generally the same approach, but a bit simpler. Here, I didn't have predictive habitat, but actually mapped habitat polygons. So these are the habitat layers I was provided from the species distribution model for Dendrophilia conifera. On top, the habitat suitability layer, below the model uncertainty layer, each classified into high and low, being 1 and 0. And this layer that combines the two, which I therefore chose as the basis of my risk assessment, it therefore has 4 categories, which are combinations of high and low habitat suitability versus high and low model uncertainty. The fishing effort, the fishing pressure layers are derived from VMS and logbook data using mean effort of the most recent 5 years available. This fishing effort was then classified into effort categories. For bottom trawling, I used the precautionary presence-absence approach. For gillnets and longlines, I adopted thresholds from Punzone al., a paper from this year on the same species and region. These thresholds represent relative fishing intensity because ecological thresholds are not yet available for the species. And then these are the categorized fishing effort layers that form the basis of the risk assessment. And individually, I crossed each of them with the habitat layer. And this is what it looked like. Sorry. Combining the categories of fishing effort, habitat suitability, and model uncertainty, I derived different risk levels per gear. So for the strict approach for bottom trawling, I received only no risk or directly medium and high risk. For gillnets and longlines, you see it's a bit more differentiated. I received no risk or low, medium, and high risk. And in comparison, you also see that longlines required more fishing effort to reach the same risk level as gillnets because they are considered less damaging. Let's look at the results. For bottom trawling, substantially overlaps with predicted suitable habitat, but I'll discuss later why this should be interpreted with caution. Gillnets showed the greatest potential interaction with Dendrophilia habitat. 7.3% fall into the high-risk category, and you see the hotspots here in red, especially in Western Galicia and of the major cities and fishing ports, A Coruña, Gijón, and Santander. Longlines, on the contrary, were associated almost exclusively with low-risk areas, and no habitat fell into high risk. For male beds, let's look at the distribution. The majority of beds are found in the southern rias, with the largest beds found in Ria de Arusa. Looking at the habitat, the distribution of the main pressure, you see that the bateas are also mainly concentrated in Ria de Arusa. Now, to account for indirect impacts, so like sedimentation or organic enrichment, I applied a 500-meter buffer around the bateas polygons. And this is the step in between, and the results show that more than a third of the habitat overlap either directly in red or indirectly in yellow with mussel aquaculture. For the cumulative pressure analysis, you see that habitat overlaps with up to 3 pressures simultaneously, and that these cumulative pressure hotspots are also concentrated in Ria de Arusa. Here, I specifically analyzed habitat already exposed to bateas and found that almost 70% of that is exposed to at least one additional pressure next to the bateas. This was a calculation, so it doesn't show in the map. And now let's discuss these findings, the key limitations, and and management implications and that I could derive from these findings. So the bottom trawling overlap is very likely overestimated due to limitations in both data sets. First, the lack of a rocky substrate layer in the model, and second, the fishing layer resolution. One cell is still 1 square kilometer big, so we most probably have a coexistence of trawling and habitat in many of the cells without direct interaction. Gear nets showing the greatest potential habitat interaction supports need for research focusing not only on trawling but also on static gear impacts. And long lines may actually be compatible with coral conservation goals in many cases. For MEAL, the key result was that they face multiple overlapping anthropogenic pressures. So this means that the cumulative effects may actually be more relevant to the habitat than effects of individual pressures alone. And next, besides the bottom trawling limitations, there's a couple more limitations I want to highlight. That is that research on static gears is still emerging, and that is also the reason that ecological thresholds for Dendrophilia conifera do not yet exist. Therefore, if I was to change the thresholds in my analysis that are now statistical thresholds, my results would also likely change. For male beds, the key limitation that I want to focus on is the lack of small-scale fisheries data, including such data that doesn't yet exist, would improve the analysis a lot because it would more realistically represent the fishing activity in the RIAs that better than my VMS data did. For example, scallop dredging that is pictured here, a serious threat, traps and other bottom contact fishing on a small scale level would be included. This brings me to the management implications. For Dendrophilia conifera, the key management implication is that it should be more gear-specific. There should be more gear-specific conservation and management measures, other than treating all bottom contact gears the same, which is currently the case in some of the regulations, and thereby also prioritizing gear net hotspots for conservation, monitoring small-scale fisheries, and improving coral mapping. For male beds, avoiding additional aquaculture pressure on the mapped marl beds by moving the activity, for example, should be avoided. Also here, the monitoring of small-scale fisheries, and lastly, important, introducing a habitat monitoring on a long-term basis to also study the actual ecological impacts, that especially in the cumulative pressure hotspots. And to conclude, this risk assessment did not only show which pressures pose the greatest risk or potential risk to each habitat, but also where. Both analysis and results ultimately support more pressure and habitat-specific conservation and marine spatial planning. And these 2 case studies also demonstrate that there is no single workflow for habitat level risk assessments, but if the analyses are adapted based on the data available for the habitat and the relevant pressures, it is well possible to apply these workflows to other benthic habitats. And thereby this work contributes a foundation is a foundation for future assessments of vulnerable habitats. Thank you for listening.
Corina, we can start with Aziz and then Steve for the questions.
Thanks very much. Good presentation. I have a question. Right at the start, you mentioned about habitat complexity and how they obviously contribute to this. So is there any way to include this in your kind of spatial analysis to see where the highest complexity occurs in association with anthropogenic pressures, including fishing?
Well, since the model is predicted, we don't have actual proof of where these corals are and how, like the density, you mean, I guess if we're talking about complexity. So, I mean, I'm considering Dendrophylla conifera as a habitat because it's a habitat forming coral, but in fact, we don't know near where they are, how big these colonies are they just individuals? And that was the reason I included bottom trawling, because we wanted to account even for single individuals that may occur in soft bottoms. But yeah, with the risk assessment, we cannot say the specific ecological details nor the impact.
Okay. And so this is the predicted model, but is there any historical occurrence data that is available that we know their distribution and how it has changed over time and how it may be compared to your model?
Yeah, the paper from Punson that was published this year, that was on Dendrophylla conifera, they actually studied the ecological, so they actually sampled and detected the habitat. But yeah, it was a completely different study because they could only speak for a small region where they sampled. And the goal here was to cover the whole NOR, and therefore we had to rely on the predictive data. But yeah, this exists, and sorry to answer more specifically, that this is why I derived these thresholds, because I figured that this is the closest I can get to current science on how, because the problem is how much fishing is too much for this habitat, and this is the most specific paper on that. Yeah.
Okay. And then lastly, you spoke about the cumulative impacts, and am I right in saying you kind of sold fishing as the main kind of impact, the impact of fishing, but there are other cumulative impacts?
No, the bateas, I mean the mussel farm.
But I mean the weight of them, like what is the largest kind of pressure?
Yeah, this is a limitation that I skipped kind of quickly. The fact that this marl analysis, it was a very first approach, and surely this can be improved, because what I did here, the pressures, first of all, I took them into account in presence absence, so I didn't for Dendrophilia, I classified the fishing into effort categories, which here I had 7 different pressures from different sources, different scales, different units. So it was impossible to, include the intensity. And for the cumulative pressure analysis, the same. I simply counted the overlapping pressures without weighing the intensity.
Okay.
And I have seen studies, again, I have seen studies where they did this for Posidonia in the Med, and this is a 3-year ongoing study for cumulative pressure analysis. So it's something that's possible, but in more time.
And finally, final one. Outside of this, what's the trends in the distribution of both these systems? Like the general trends, are they declining or are they stable?
I mean, both of these habitats are included, were included in the BioDiff, which was a 4-year-long European project that will actually continue soon for a reason, and they are considered by all these frameworks. But it's true that for MERL, it's very specific. They are specifically listed in OSPAR, in the Nature Restoration Regulation. Dendrophilia conifera, and sorry to finish on MERL, yes, they are decreasing. The last assessment or kind of monitoring has been 2010, and that's why it was so important to do a reassessment. And it is shown that this habitat is decreasing, has been decreasing since the pressures increased since 1950s. But there has been no long-term monitoring to really show the changes. So this would be the next step. And for Dendrophilia conifera, in comparison, they are quite widespread. Also due to the conditions along the whole Nor. It's a rocky shelf, so they have plenty of habitat.
Substrate.
Exactly, where in the Mediterranean is much more a carpet of.
Thank you.
Okay, that's this. So can we just
Thank you. Yep. Thank you for your presentation. And yeah, you're right, actually. Cool. Especially the calcifying one. So why do you think that mussel farming is bad for mermaids? What would be the effects?
Yeah, if you see, I think I had a picture included here. This looks quite idyllic, but if you are there and you look at it from the air, there's more than 3,000 of these platforms in the few areas. So this is few square kilometers actually, and so that it's very, very concentrated. They first of all, they take the light away from the habitat. So the habitat is the beds are very shallow, between 5 and 25 meters. So it's very the habitat is around and halfway under. It's actually hard to say because they couldn't sample under the bateas. Anyways, they may have died already under because there's no light and there's a lot of organic substrate that goes into the water as they process the mussels. I have seen how they come with boats and cranes and they take out the big ropes and clean everything right there. So they're very sensitive to the changing water, the sedimentation, and this organic enrichment.
Okay, yeah, because we could also think that mussels kind of clean the water in some ways, and that would compensate a little bit for it, and algae would have less particle in the water, which they might prefer the mermaids, maybe.
Yes, if then the boats didn't come to. I guess, to know for sure, and this is something I had the habitat condition data available, but I did analysis around it, but couldn't statistically find any clear trend because I tried to find out whether more pressures lead to a more degraded habitat. And in fact, what I found is just that in this area under intensive pressure, you have very good quality male beds that need to be protected.
Okay. I have a question on the species distribution model that you used. It's based on what, this model? Which, what do they use to say this place is suitable or not?
They included a few actual coral occurrences, and then all the environmental variables. So the slope and all the environmental variables known from literature on suitable dendrophilia habitats. The only thing that wasn't included in the model, which is one of the limitations I mentioned, is the rocky substrate layer, because the layer currently available on EMODNet is not reliable at this resolution that the model was.
Yeah.
And that's why it wasn't included at all. Does it answer your question?
Yeah. Okay, thanks.
Thank you, Steve. We can go back online with Kayla and
Thank you. Thank you for your presentation. I have a question, because so if I understood correctly, the model is based on coverage and presence absence. Am I right?
The species distribution model for Dendrophilia conifera.
Yes.
It categorizes the habitat suitability and also the model uncertainty. So how sure is the model about whether this area provides suitable or not suitable habitat?
Okay. And so there was no inclusion or consideration into how the different populations or different sites could be connected with each other. So how the dispersion of the corals could be affected, right? Meaning by, for example, if you have in between two zones that are suitable, maybe there is a flux of individuals because they reproduce sexually, and maybe if you're fishing in the middle, you can disrupt this connectivity. Is there a way to include that, or was it included?
No, I don't think so. Those are all ecological aspects you're mentioning that are really important, and I guess the only way to know better is to do ground truthing with the results that I've received now. So in the model, that wasn't possible to include, and the risk assessment is an assessment of risk and not the ecological state or aspects.
Yes, but if you're considering the risks, couldn't also be included, like the risk of impacting the connectivity between the different populations?
If I had what I did is that I overlapped habitat polygons with pressure polygons and or pressure polygons or pressure data. So if I had data for this, yes, but I guess a first improvement would be an improvement of the fishing data before, I guess, the connectivity is interrupted where we have a lot of fishing, like, for example, bottom trawling within different rocky habitats, right?
Yeah. Or also because if you're destructing the rocky bottoms or any bottom where the coral could settle in between, it also disrupts and could also potentially affect the colony, even though it's not directly where the coral is. It could also be a place where it could be.
Yeah, and I mean, the only way I tried to incorporate that was by including bottom trawling, because I considered this connectivity that if these individuals, okay, and this is much more relevant in the Mediterranean, where you have less rocky habitats, especially less connected rocky habitats, and that is more important to keep this connectivity alive. So including bottom trawling to even protect or detect potential individuals in soft sediments. But in the north, where you have rocks all along the coast, the connectivity is not such a big problem as it is for the Mediterranean.
Thank you.
Thank you.
Thank you, Kayla. And so we're coming back with you, Emma.
Thank you, Christophe. Thank you for your presentation. I have my first question concerns the fishing intensity. What do you consider fishing intensity?
This is the raw data that I used, the layers. So I had data from 2009 up to 2023, and for the static gears to 24. And this is the raw data for bottom trawling is measured in swept area ratios, and for the static gears in hours per square kilometer. So that's the how fishing intensity is measured. And then to avoid interannual variability, I used the mean fishing effort. And then, yeah, I guess the difficulty was knowing how much fishing, regardless of the values, how much is too much for the habitat, like where do we cut low effort, moderate effort, and high effort?
Yeah.
That's your question, right?
Yes, that's my question. I was wondering, why didn't you use the CPU, the catch per unit of effort, which can be suitable also because you have different fishing techniques, you know. Have you thought about it, or is there a reason why you didn't use it? I'm not saying it's the best.
Well, maybe I can answer the question by explaining how this data was gathered, because it was a proxy of speed. So this effort is already considering how fast the vessels are moving. So depending on the speed, it's really a proxy. Are they fishing? Are they not fishing?
Ah, okay.
So that's the best
VMS,
available fishing
data, right? Okay.
Yeah, layers that are available. In fact, I changed the layers throughout the process because there was more recent data and that better resolution available.
Okay.
And then this is really the best I had.
Okay.
And then these are the thresholds that were used for the same purpose. So even though they're not, I mean, perfect would be ecological thresholds, meaning you have a pressure response threshold. And this is under progress. This has been researched by colleagues at IEO, currently for bottom trawling, but not yet for the static gear. So it didn't solve my problem for now. It will pretty soon, hopefully.
I like the for now.
Yes.
It will be solved somehow. My other question is about the fact that with the gill nets and the long lines, there are no VMS. You spoke about the bias in the data. How did you manage that later on your in your methodology? Like, it's very good that you highlighted it. Like, I cannot agree more, because it's the case, and it's a problem when we want to do this kind of assessment, that these vessels are artisanal, like small scale fishing. How did you manage these 90%? Have you thought about an indicator, or it's just you did with the 10% left and that's all?
In fact, I can't even say it's 10% because it's not really black and white. So I have the number of vessels in the statistics. I can see how many vessels are more than 12 meters. So in the whole NOR, it's around 60 per gear type that we have data of. And then some of the small, so gill nets and long lines, some of them may be larger than 12 meters, some may be smaller. And most of the most of the small scale fishing vessels stay closer to the coast, which relevates a little bit this limitation, that it's not as big or as important as I first thought. But I tried to get a clear percentage, because I wanted to know by how much is it underrepresented, and I couldn't even tell you. It's not 10%. It's actually less. And but yeah, the next step, the only step to really know would be to equip the small scale vessels with any type of GPS, which for Cantabria and Asturias is in implementation since 2024. And we've received the first data in May, but like first preliminary data, so it's far from being ready to use. I couldn't use it for that, but that would solve many of these issues. And for Galicia, unfortunately, it would be very relevant for the RIAs, as I said, This is not even planned yet.
Okay.
And so these results, at least they give a good indicator, because I mean the risk, it's real, even though it's underestimated. But I mean it's a better than nothing for now, and maybe a good reason to argue for GPS tracking monitoring of small-scale vessels.
Yeah, totally fair.
Mm-hmm.
Maybe my last question would be about the implications of your work. If you were, I don't know, the mayor of Galicia, for example, what would you do with these results? Like, what would be the next step? Would it be an implementation of a protection status for those species? Is there already some protected areas there? How do you see next step, if there were any?
To be honest, it wasn't on a. Sorry, I wanted the slide. It wasn't on such a regional level, but the EOS, the National Research Institute for the Government. So this is where the final results of the BioDiff project of this 4-year-long period were presented at the ministry in Madrid. So these results that I had were presented there and included in the reports. In fact, they attached the whole master thesis. And it's really up to the national government to take this into the EU Commission, etc., and release regulations or even change existing regulations. In my thesis, as you know, I suggested measures as good as I could. But I mean, these are recommendations, and it's up to the national government to implement them.
Okay.
These are protected species, right?
Yes.
Yeah, so they will be under the NRR, the nature restoration.
Yes, exactly.
So your work is good for where to prioritize restoration, possibly, and to avoid anthropogenic impacts.
Definitely.
Yeah.
Yes, exactly.
Well done.
Thank you for your presentation. And I wanted to ask you if you think that it's possible to collaborate with the small fishing vessels in order to have some data on the bycatch of the bottom trawling, if you think it's possible.
I know that some people from EO have good relations with the local fishermen. It's kind of on a team basis. I know that they accompany them and that they do research through these vessels. One suggestion of mine was to implement the reporting of coral bycatch, yes, through all of the gears. This is currently not in place, and I guess because here I'm talking about the larger vessels, it's more difficult to approach a bottom trawling vessel than to work with small-scale fisheries. And the truth is that they're not interested in this GPS tracking. Yeah, and the only other source for this kind of bycatch data for now was EO's own surveys that they do annually, the MARRES surveys, where they use the same gears. But yeah, it would be an approach. Sorry, just asking.
I have 2 small questions. You mentioned logbook data. What does that refer to?
I guess in addition to the VMS data, because all the boats have logbooks.
Okay,
Captain reporting.
Okay. And then if no one else is, what does it mean also when habitats you mentioned overlap indirectly with the aquaculture farms? What is indirect overlap?
This buffer zone of 500 meters that I applied, and this was based on several literature, because the actual overlap in the, it's worth going back. the actual overlap is small, you see. You see the direct overlap is rather small. You would expect it to be bigger, but I mean, several reasons can explain that. For example, that, as I said, they couldn't sample under the pateas. The habitat under the pateas may already be degraded, so we don't have a comparison, and historically they have been under pressure. And so since this direct overlap is very small, and knowing the impact of not just direct lack of sunlight or, but all the organics and sediments that are carried with currents and the tides, I applied this 500-meter buffer, and this was in line. Here, this is the 500-meter buffer. And thereby you have a direct and indirect overlap. And for all the other pressures, and this was one of the biggest challenge to find justified buffer zones, because for contamination, I had points where the pipe of wastewater treatment plants entered the sea. So clearly I wouldn't have any overlap. And for that, I applied a 1-kilometer buffer, also based on literature. Same for dredging, for example. Mm-hmm.
Oh, good. Congratulations, Corinna.
Thank you.
I would like to take this as an opportunity to thank your supervisor, Nina Larissa Arroyo Alberto, and Jose Manuel Gonzalez Rusta.
Yeah, I think they're listening.
Is are listening?
Yeah.
So thanks to you from your CSIC Santander. I hope I haven't mispronounced your name too much from Spain. And so as Andrea is coming here, also like to thank your supervisor directly, David Abekassis and Elisa Prakopoulou from the CCMAR at Algarve University in Portugal. Maybe you might be interested me changing the slides.
Yeah. Okay.
But if you want,
Let me
you can try.
Yeah.
Yes. Oh, good.
Yeah.
Just scroll down. And I think we're all fine.
Okay. Shark nursery areas occur predominantly in high productive coastal ecosystems, the same ecosystems that are highly used by fisheries, which is why in most regions, the knowledge that we have about nursery areas comes directly from fishery landings. And this creates a conservation paradox where we're getting to know these high vulnerable areas through their mechanism of exploitation. Meanwhile, in other regions with higher conservation funds, they have specific programs to study these sites. So here a question is raised on how can we use the already available information to get to identify and study in these nursery areas in data poor, underfunded, and even difficult to access areas without relying on fisheries. This is why I decided to use the species distribution models to understand and study the global habitat suitability of the nursery areas of the scallop hammerhead and the black-tip sharks in order to understand the ecological drivers, future climate change implications, and overlap these results with marine protected areas to understand their current level of protection. My name is Andrea Vera, and I developed this thesis under the supervision of David Abekassis and Elisa Francopulo at the CSMAR in Portugal. And sharks, during their first weeks when they're born, they actually suffer from really high mortality rates due to inefficient feeding and really high predation, which is why nurseries are these habitats to provide enough food and protection to allow their optimal growth. Yet, not area that has small sharks is considered a nursery. Under the latest framework for an area to be considered a nursery, the newborns and young of the year present in the area must have higher abundance compared to surrounding areas. These individuals, they also need to present long residency periods, and this area needs to be used consistently over the years. And our species, the scallop hammerhead, is a critically endangered species. The black-tip shark is a vulnerable species with decreasing population trends. Both exhibit similar global distribution. Yet, more importantly, these species have some of the most well-known nurseries around the globe, yet they're concentrated along the eastern tropical Pacific and the northwestern Atlantic, which makes them excellent model species in order to apply a species distribution model to bridge this gap between known nurseries and the ones that are still under-identified. Which is why the main objectives of my thesis were, first of all, to create this global baseline of the nursery areas and their ecological characteristics. Then, with the use of species distribution models, understand the key environmental drivers that guide habitat suitability for both species nurseries, and then to predict potential nursery habitats globally and understand the future implications for conservation. So for this, I conducted a literature review using Scopus as my main searching engine in order to identify the nurseries, the location of these nurseries, and the literature and the environmental characteristics highlighted in literature to influence this nursery selection. I also ran a backward citation research and looked into other sources that have already identified these nurseries. And I complemented this information with important shark and ray areas that have been categorized under the subcriteria C1 of important reproductive areas. And with the polygons provided by the IUCN Shark Specialist Group, I used QGIS in order to extract random points from these polygons in order to use them to develop the species distribution models. So, to develop a species distribution models, what we need to understand is that they use a species occurrence data and environmental data in order to infer global habitat suitability within a specific region. So from our literature review, we already have our known nursery locations, and for the environmental data, I extracted the data, these environmental layers from BioOracle, that is this global comprehensive data database with layers specific from modeling and from the environmental characteristics highlighted in literature that are important for the nursery selection for both the species. I use the minimum and maximum temperature, minimum oxygen, salinity, minimum productivity, and diffuse attenuation as a proxy for turbidity. Then I develop and assemble species distribution models using 2 machine learning algorithms, boosted regression trees and extreme gradient boosting, and a more traditional approach, the generalized linear models. And I follow the workflow provided by the biodiversity distribution modeling course that's publicly available, and it has been produced by George Assis. So, in order to bridge this gap of the data that really don't have any information about nurseries and these highly localized studies, I projected these results globally within each species known distribution. And from these results, in order to understand the future climate change implications, I projected these results into 3 contrasting end-of-century climate change scenarios using the shared socioeconomic pathways that you can see here, that they have increasing emission tendencies. And in order to understand the area dynamics between the suitability of the present day and the future climate change scenarios, I contrasted this suitability in order to identify future loss, gain, and ecological refuge zones. And finally, to understand the level of protection granted for these areas, I overlapped these results with the marine protected areas layers that were extracted from the World Database of Protected Areas. And I overlap them with the identified nursery locations that are the red dots and the different predicted suitable habitat scenarios. So our final database of our nurseries from the publications that met the inclusion criteria and from the ISRAS included a total of 113 nurseries for the scallop hammerhead and 76 nurseries for the blacktip shark, including the primary and possible nurseries and ISRAS. And these locations were used to develop our species distribution models. So what does this model say? So turbidity was found as the main driver to guide nursery habitat selectivity for both the species, followed by minimum oxygen. And minimum productivity was found the least important factor for habitat selectivity for the scallop hammerhead. Meanwhile, maximum temperature was the lowest factor to influence habitat selectivity for the blacktip sharks. And this actually aligns with literature that highlights how turbidity acts as the main mechanism for habitat selection, as it acts as a predator avoidance mechanism and also to improve predation. And in some papers that have studied dissolved oxygen concentrations, they have seen that there is a selectivity of lower dissolved oxygen areas in nurseries compared to non-nursery areas, and that this is actually a limiting factor for habitat use for the nurseries. Then when we look into the predicted habitat suitability areas, this extends for around 4 million square kilometers for the scallop hammerhead, concentrated mainly along the eastern Indian Ocean and the northwestern Atlantic. And for the blacktip shark, this extends for around 2.4 million square kilometers, concentrated mainly along the western Atlantic Ocean. And when we overlap the known nurseries that are these black dots with our suitable habitat, predicted suitable habitat, we can see great disparity among regions. And this highlights how we can find new active nurseries, mainly along the West Africa, that you can see there are no dots, no information, and also along the Southeast Asia, mainly for the blacktip shark. And we can also see how the Indian Ocean and the Western Atlantic are some other regions with potential of finding even more active nurseries for both species. Then, when extending our predictions globally, we can see a great area contraction for both the species across all scenarios with increasing tendencies as emissions increase. And for the scallop hammerhead, the greatest contraction was actually found under SS2, reaching the highest suitable area contraction of around 13.5%, which was greatly offset by the creation of new habitat, that is the yellow area. Yet this area loss was actually disproportionately larger for the blacktip sharks, which by the last scenario, SSP3, it reached over the 50% of suitable habitat loss, with very minimal habitat gain that was actually concentrated along the Peruvian coast. Then, when overlapping our results with the marine protected areas, we can see that very minimal protection is actually granted to this predicted suitable habitat, being around 15% for the scallop hammerhead and 12% for the blacktip sharks. And looking deeper into these MPAs that grant this protection, only around 27% of them and 15% respectively are MPAs that have been designated as partially or all no-take zones. And yet, when we look into the present-day nursery records, these values increase a little bit. 38% of the present-day nurseries for the scallop hammerhead are currently protected within MPAs, and 53% of the blacktip sharks are protected under MPAs, which are these purple MPAs that you can see in both maps. And when we mix both results, the future scenarios, current protection, putting as an example the highest emission scenario considered, we can see that some nursery areas for both the species fall under future predicted loss areas that are actually currently within NPAs. And on the other hand, we have other nursery areas that fall under these future climatic refuge zones, which is very good. And at least 40% of them fall within MPAs, but there's at least these 50% of MPAs under future refugia that are not currently protected. So all of these results actually make us realize that we need to take proactive and adaptive management measures in order to adapt to this dynamic nature of the climate and of the nurseries, and to reduce the recurrent anthropogenic pressures for these areas. And it also leaves us with some questions on how should we manage these protected future lost areas and these unprotected future refuge zones. So finally, I want you to remember that historically, shark nursery areas have been known where sharks are caught. Yet, this thesis highlights how we can use ecological knowledge and the species distribution knowledge in order to pass this limitation and study the nurseries without relying on fisheries, which makes it an scalable and cost-effective framework to guide future surveys and conservation actions and measures to study the areas that have the least information. Thank you so much for your attention, and I want you to leave you with this message that if we want healthy oceans, abundant fisheries, and thriving coastal communities, we need global efforts and stronger regulations in our MPAs, more importantly, as we're reaching our 30 by 30 goal. Thank you so much.
Thank you, Andrea. We will go first with Emma and continue with Kayla.
Thank you very much for your presentation. I enjoyed it.
Thank you.
Yeah, thank you also for putting the photos of sharks reminding for each species. That was really useful to follow. I have a question regarding the modeling you did.
Yep.
So, according to your results, the turbidity of water is a more driver for habitat suitability than the temperature itself.
Yep.
That's quite uncommon. Usually, it's the temperature. So, would you say that sharks would not be affected by the temperature rise in the oceans, or is that a result that makes you think of other explanations? I don't know.
Yeah, that's a very good question. And yeah, if we can see here, for example, maximum temperature was, yeah, well, first of all, I did this ensemble model, right? So each model had maybe like different outputs on the importance of each factor. And for example, we can see that maximum temperature for Spirulina lewini has a great variation in its importance, and it's because each model maybe considers the different influence within its model, right?
Okay.
But I would say honestly, is a combination of factors.
Okay.
Yeah, it's a combination of factors, and I would say for sure future climate change will impact the species. Indeed, I didn't mention it here, but I also did the species distribution models for the general distribution of the species. And for them, temperature was a great factor.
Okay.
So if you combine the impact of the general species and actually it showed these like latitudinal change in distribution, it will have this indirect impact.
Okay.
Temperature, yeah.
Okay. Thank you for that. My second question is about the habitat itself. Did you somehow combine your results with a habitat layer, even if the resolution is not very fine, to see if in the habitat that is reported in literature of these sharks are well overlapped with your results? Because sharks rely very much on the habitats itself. So,
yeah.
Honestly, no, I didn't overlap these with the habitat layers.
Okay.
I more compared like the general distribution of the species with these results. Yeah.
Okay. Great. Thank you.
Yeah, you're welcome.
Thank you for your presentation. Very interesting and a lot of work. So congrats. So from the beginning, I'm going to continue on the environmental variables you choose. So from the beginning, you chose to focus on oxygen, temperature, turbidity, salinity, on these specific variables.
Yep.
Why?
Yes. So as I mentioned, you're asking why did I choose these variables, right?
Yeah. For example, why not also including, I don't know, pH or?
Yeah. Well, as I mentioned, I did this literature review before, and I gathered all the variables that have been studied in literature, and indeed I didn't include all of them. I include the ones that have been highlighted in literature already as important factors for habitat selectivity for these species, and also for a species distribution model to perform the best. Like you don't need all of the variables. You actually need to choose specific variables that will guide your model. Otherwise, you have too much confusion, and maybe your results won't be showing the reality of the ecological nature of the species. But the simplest answer is that they are based on the literature review that I did previously. Yeah.
Okay. And I agree with that. It's pretty surprising that you found the temperature not being the primary driver of it. Did you try to consider other temperature variables, not just the mean? For example, the variation or the intensity of different heat waves or this kind of stuff?
Yeah, honestly, something that I would have loved to do is I've seen other papers doing it, is the seasonal species distribution models. As the nurseries, they're actually seasonal. The habitat use, like, okay, so sharks they use these nurseries seasonally. So they, in most areas, they come like for 6 months and then they migrate or they move. So I would have loved to do this seasonal species distribution model, but the layers from BioOracle, they are actually decadal data. But I actually use the long-term minimum and maximum. So it's the average of all the minimum temperatures of all the years and all the average maximum from all the years, which is a good proxy as well. Yeah.
Okay, thank you. And did you those nurseries locations? Do you have information about the concentration of prey as well, or that would be a further step, for example?
The concentration of prey, yes, that would be like pretty interesting. Yet, for example, in many studies, they have already found that prey abundance is not that important in some areas for habitat selectivity, as for instance, in Hawaii. Actually, most of the mortality rates of the sharks there are due to starvation. So they have seen that actually like primary productivity is not relevant. So, and this is maybe something related of why turbidity was so important, and it's because in different areas they have seen that protection, like predator protection, is the main driver for habitat selectivity, and turbidity is kind of like this proxy for predator avoidance. So, yep.
Okay, thank you.
You're welcome.
Okay, that's we can continue and we will finish with SS.
Thank you. That was very interesting. I was wondering, like, do you think your model might be there may be like a bias because you are, using data where your nurseries are. For example, like, you say temperature doesn't have a big effect.
Yeah.
Is it because all the nurseries are known to be in places where temperature doesn't vary much? It's more tropical areas.
Mm-hmm.
But actually, temperature has an effect if it's outside of this range where nurseries are already found. You think this could have an impact?
Well, if you mention bias, for sure there is bias. As you can see, actually 80% of the data comes from the Western Hemisphere.
Yeah.
And well, I have the data, like the, wait, I'm gonna show you. Okay. Yeah, here. So the performance, if you see the performance of the nursery models with the AUC sensitivity, very good performance, and it lowers in boys that it actually see how well the model is predicting the actual presences of the nurseries. And I was reading, and I have it's not a bad metric, it's a good metric, but it's because there's this really high concentration in one hemisphere. And as there's also this suitability in this other hemisphere, the model kind of like identifies this like imbalance in the data. But I wouldn't say there's a bias in the temperature, as if you see also the distribution, you have very also like latitudinal, like it's not only concentrated. You don't only have in nurseries within the tropics, which maybe there you could say, okay, they have like this stable environment. You can also see a lot of nurseries in more latitudinal areas that can give these like variations. So yeah, I wouldn't say there's this temperature bias.
Okay.
Yeah.
I don't remember my other question.
You want to go to Aziz? We're coming back
to you.
Yeah. Thank you very much.
You're welcome.
I know that this is like a fraction of what you actually did.
Yes.
So I was really impressed by what you did, because really you did like 6 papers worth of stuff, and this, it was way too much. So, my first question is, what is the total known number of nurseries worldwide?
Yeah. Well, as I showed before, honestly, if you see a lot of papers that talk about nurseries, they really mention like a quarter of the ones that I found. But for instance, for the scallop hammerhead, they're like 116, the one that I identify. But the ones that have been identified as primary nurseries are the ones in blue. So mainly along the Eastern Tropical Pacific and some in Florida. And the same happens to the black-tip sharks, which actually the primary nurseries are concentrated in the Gulf of Mexico and Florida.
Yeah, so how many of these are under protection or fall within an MPA
now? Yeah. So, well, for instance, wait. Okay, some of them. Like, for example, these ones are some examples of the So nurseries that fall within like future refugee zones that are under a current MPA. So for instance, Galapagos, we have in Central America, the Gulf of Mexico, here in Australia, we have in Tanzania, South Africa, for the black-tip sharks, Galapagos, Gulf of Mexico, and Australia and Fiji. Mm-hmm. Yeah.
And in your model, these specific locations are also predicted to lose.
Yeah. Well, yeah. And for example, this is like the interesting part, that these are specific nurseries that are within future climate change, like future area laws, like suitability laws that are currently under MPA protection.
Yeah. And what is that? What do you think that loss will be driven by?
Well, it can be mainly temperature, and then all the all the environmental factors changing. And for example, if we find that turbidity, well, turbidity was more a stable variable because there's not a future prediction of turbidity. So would you use the present day turbidity? So it would be mainly the minimum oxygen and like in order of relative importance.
Okay. Slide 26.
Yep.
Sorry, it's a bit far.
No worries.
You highlighted in the presentation like this is the total suitable habitat in the SSP1, SSP2, SSP3.
Mm-hmm.
But it increases, right?
Yeah, it increases here.
There's a rebound.
You mean the suitable area or the...
Yeah, the total suitable area goes decline and then increases.
Yeah, it declines and increases for the last one, but because you have more habitat gain. You have like more creation of suitable habitat in the future. So these are like new areas that will be suitable in the future for these species. That's why I mentioned the highest like global loss, not only considering the present loss, but considering the future area, created area. The greater loss is under SSP2 and not in SSP3 for the scallop hammerhead.
Mm-hmm. How do you interpret that result? Like, you know, this is the...
Honestly, if you see the distribution and the creation of the habitat is mainly in the Persian Gulf. So honestly, it's not like, oh, wow, like a lot of area is created worldwide. It's like a very concentrated area. But yeah, honestly, it, like even I would more interpret it in the way in which the known, like how close our nurseries, like our present day nurseries, are in near future loss areas rather than other way.
Yeah. What impact does this have on MPA design? You know, the investment going into these things, the planning.
Yeah, we were talking this like with Elisa, and it's like okay, like should you continue fund like an MPA that is in actual like future loss areas, or like change that like investment into like future refuge zones? But that's why I mentioned that we should consider these proactive and adaptive management measures. So I would say that first of all, we need to identify more nurseries, but for the ones that we already know and they have the budget to do it, I think there should be like a current like monitoring of the area and the environmental characteristics of the area to kind of understand if they're actually being actually know if these areas are being currently used or they're stopped being used and maybe move to other suitable areas and be a bit more adaptive in this way. Yeah.
Okay. Yeah.
Steve, yeah, I just had a question on the bioreactor models that you use.
Yeah.
So what's the resolution of those models? Like, because you might be going very wide, while the nurseries are very localized in small bays.
Yes, that's another limitation, but it's not a real. I wouldn't say it's like a very huge limitation. I discussed this with Elisa, with my supervisor, and well, BioOracle, it has a resolution of 0.05 degrees, which is around 5 kilometers along the tropics, and 9 kilometers in the latitudes. And actually, like the nurseries, they're kind of like wide, like very like spread apart. So it's like, and that's actually kind of like the nature of nurseries, you know? So it's like you have one in a very specific place because it needs to have higher abundance compared to the other areas. But I would say that because nurseries are found in these like very specific bays with these very specific conditions, having a greater resolution would highly improve the models. Yeah.
And you didn't try to look into data from specific places where the nurseries are, you know, like if there is local environmental monitoring, or like, you know, a lot of those places are near coral reefs, for example, or there's tons of temperature loggers.
Yeah. The thing is, for example, for the future climate change scenarios, that wouldn't be possible to include them.
Yeah, it was more
Yeah.
to look at.
Yes, exactly. Like if I wouldn't be like targeting these like future climate change scenarios, for sure you can use like better resolution data to have like more precise present day data, like habitat suitability maps. Yeah.
Good. So here you go for the next question.
Yes. I have maybe a silly question, but could you go back to the map of the like scallop hammerheads future versus current nurseries?
This one.
Yes.
Yeah.
I was just wondering. So of course, we know that they're migrating species that have like predictable migrating patterns, right? So
Mhm.
they always kind of go and follow the same migration
pattern. Yeah.
How do you think they'd be able to adapt if their nursery suddenly like, I don't know, it's suddenly like so much further away than where they usually go?
Yeah.
How do you predict that, if you can, I don't know.
Yeah.
That they would be able to adapt to this?
Yeah, honestly, I feel the problem now is the other way. With the global species distributions, if you see the future predictions, the habitat suitability laws, it actually increases to like 60% for the species, and they now distribute along the tropics, and you see like these like huge red band in all the tropical area. So it's like it raises this question on like, what will happen if like all the tropical area is like unsuitable for the adults and their nurseries in the tropics is still suitable? Like, where are their nurseries going to go? And also thinking that they have this site fidelity, like what's going to happen. But I would honestly say that it's a matter of ground truthing what other, yes, just like being there, like really understanding what's happening. Yeah.
Oh, you're getting tired. I have a question. If you want to come back to the gains of in the SP3, yes, on this one. So here, your if you have a look to the suitable area, you consider an area wherever it is in the world. Just looking on an ecological like thing, here the area gain, you say it yourself, is very, very specific.
Specific, yeah.
What about like considering also the effect on the genetic pool of the whole species, and the fact that if you have 15% addition, but in a very local place, it may be not so good for the whole.
Yeah, in this case, well, we cannot see, but it's actually quite spread along like North America, South America, but in like latitudinal areas, and like, and in this way, I would say there would be this maybe a bit of like well, also thinking that I only consider 2 species. If we take into account that there are these that other species have a specific nurseries, maybe these future suitable nursery areas will overlap with already available nurseries for other sharks. And like I feel like more ecologically, these may represent a bit of like competition in resources and in habitat use with other species.
Okay. Your answer is interesting, even if it doesn't answer my question. But
yeah, can you repeat then to actually understand?
It's like the mosquito parts.
Yeah.
It's so the gains are mostly in one place.
Yeah.
And my question, but maybe it's like if you have much more larger area, but only a given place. It's not going to increase the genetic pool of the whole species.
No, it's not.
Yeah.
Yeah.
Good question.
No, also there's like high genetic diversity, like there are subpopulations that they don't mix within ocean basins.
Okay, because I would imagine that the best case scenario is that we have yellows everywhere, so that's globally speaking, you have the subpopulations more genetic.
Yeah, I would say the most like important cases for the black tips, that they actually the only area considered to be like to increase this ecological suitability it was actually only along the Peruvian coast, like nowhere else. Yeah.
Okay. Thank you, Andrea.
Thank you.
You have some messages from some people that I may share.
Oh.
Also a question from Emily, but okay, coming back.
Okay, let's go.
That's good. A question from Emily that has been largely discussed also by others. Thank you, Andrea, again.
Thank you.
Okay, another one. Of course, I'd like to thank those jury members, Steve, Emma, Aziz, and Kayla. And for all of your questions, it was a little bit warmer in the room, so thank you for still asking all these questions. Next session will be still on ecology, ecological processes and interactions. We'll have Emily, Maxime, Agathe, and Lisa tomorrow morning. See you there. Bon après-midi. Enjoy your evening. Yes, as usual, I will ask nicely to you to leave the room, and please, Kayla, stay around.